Hello students and welcome. Today we will be discussing the topic titled Determination of Relationship between Chemical and Biological Data of Drugs via hash analysis. The main objectives of the present deliberation will be to introduce the various physicochemical factors affecting the activity of drugs, to introduce the basis of hash model, to understand the relationship between the chemical structure and biological activity of a drug molecule, to introduce the accuracy and success of hash model. Drugs belong to the category of chemical substances that are employed to prevent or cure diseases in humans, animals, and plants. The identification of a new drug molecule using the traditional medicinal chemistry approach requires a lot of synthesis, time, and money. It was identified that out of billion molecules synthesized, around 1 or 2 molecules reach the clinical trials. This produces hurdle in the discovery of new chemical entities, that is NCEs, for the treatment of various diseases by the traditional drug discovery process. The aforementioned facts necessitated the researchers to search for the alternate methods which determine the biological activity of the molecules without synthesizing them. Compounds with similar structure often tend to have similar pharmacological activity. However, they usually exhibit differences in potency and unwanted side effects, and in some cases different activity. These structurally related differences are commonly referred to as structure-activity relationships, commonly abbreviated as SAR. A study of structure-activity relationships of lead compound and its analogues can be used to determine the parts of structure of the lead that are responsible for its biological activity and also its unwanted side effects. This information is subsequently used to develop a new drug that has increased activity, a different activity from an existing drug, fewer unwanted side effects, and improved ease of administration to the patient. This selection of the chains required to produce analogues of a particular lead is made by considering the activities of the compounds with similar structure and also the possible chemistry and biochemistry of the intended analogue. For example, replacing a hydroxyl group with a methyl group could reduce the water solubility of the analogue and also its ability to hydrogen bond. The former could reduce its ease of absorption, whereas the latter could affect its ability to bind to its target site. Now coming to QSAR, that is quantitative structure activity relationships. QSAR quantitative structure activity relationships have been applied for decades in the development of relationship between physicochemical properties of chemical substances and their biological activities to obtain a reliable mathematical and statistical model for the prediction of activities of new chemical entities. Quantitative structure activity relationship models, that is QSAR models, have been devised time to time to establish such structure activity relationship. Chemical information and activity of a drug molecule can be inferred from the molecular structure of similar compounds whose QSAR models or classification models have already been devised. By QSAR models, the biological activity or property or reactivity, etc., of a new or untested drug molecule is predicted prior to its formulation. Having understood the QSAR concept, let's now see the correlation of chemical structure and biological activity of a drug molecule using hash analysis. Since the 1960s, enormous efforts have been made by various investigators to develop quantitative parameters to describe the biological activity. During this period, Corwin Hush and co-workers made important breakthroughs in this field of science. In 1960, Hush et al. Proposed a mathematical model correlating chemical structure of a compound with its biological activity. Hush analysis is based on the attempts made by earlier workers, notably Richardson, Richet, Mayer, Overton, Ferguson and Colander. Hush and co-workers in the early 1960s proposed a multi-parameter approach to the problem based on the lipophilicity of the drug and the electronic and steric influences of the groups found in its structure. They realized that the biological activity of a compound is a function of its ability to reach and bind to its target site. Hush proposed that the drug action could be divided into two stages. First, the transport of the drug to its site of action and then binding to its target site. Assumptions of Hush analysis are as conformational changes that take place in the target site can be ignored. There is no metabolic interference in drug transport. Linear free energy terms are relevant to receptors' affinity and additive in nature. Relationship between potency and lipophilicity is linear or parabolic. According to Hush model, the rate limiting step for many biological responses to chemicals can be defined in simplest and most general way as a random walk from the point of administration to its site of action, in which the molecule in question makes its way from a very dilute solution outside to a particular site in the cell. This step is considered relatively slow process, the rate of which is highly dependent on the molecular structure of the compound in question. This model assumes that for many types of biologically active molecules, there will be one key rate controlling reaction at the active sites. Let's put this in mathematical form as rate of biological response, that is d response upon dt, is equal to ACKX, where A is the probability of a molecule reaching a site of action in a given time interval and C is the extracellular molar concentration of the compound being tested. The product AC represents the effective concentration at the sites of action. The constant Kx might be either an equilibrium or rate constant. It's assumed that a relatively large number of reaction sites are available so that these remain essentially constant during the test interval. Also, during the drug transport in the body, it has to pass through the numerous membranes, and so the ability of the drug to reach its target is dependent upon its lipophilicity. Consequently, this ability could be expressed mathematically as a function of either the drug's partition coefficient or pi value of appropriate substituent. Partition coefficient in pharmaceutical sciences is used to estimate the partition of drugs within the body depending upon the degree of hydrophilicity of drugs in different phases. Hush's reason for taking partition coefficient is that for Nutella cells, the rate of movement of a great variety of organic compounds through cellular material has been found approximately proportional to the logarithm of their partition coefficients between an organic solvent and water. However, on reaching its target, the binding of the drug to its target site, besides its degree of lipophilicity, depends on the shape electron distribution and polarizability of the groups involved in its binding. A variety of parameters are now used to describe each one of these aspects of drug activity, the most ones being Hammett's electronic sigma and the Taft ES constants. Therefore, the model is a steady state model. Hush postulated that the biological activity of a drug could be related to all or some of these factors by simple mathematical relationships based on the general format. That is, log 1 upon C is equal to K1 partition parameter plus K2 electronic parameter plus K3 steric parameter plus K4, where C is the minimum concentration required to cause a specific biological response and K1, K2, K3 and K4 are numerical constants obtained by feeding the data into a suitable package. For example, the general equation relating activity to all these parameters often takes the general form as log 1 upon C is equal to K1 into P minus K2 into P square plus K3 sigma plus K4 ES plus K5, where other parameters could be substituted for P, sigma and ES. For example, pi may be used instead of P. However, it's emphasized that these equations, which are collectively known as Hush equations, do not always contain the main three types of parameters. The numerical values of the constants in these equations are obtained by feeding the values of the parameters into suitable computer package. These values, that is pi, sigma, ES, are obtained either from the literature or are determined by experiment. Many QSAR investigations involve varying more than one ring substituent. In these cases, the same parameter for these substituents are expressed in Hush equation as either the sum of individual parameters or independent individual parameter. For example, in the hypothetical case of a benzene ring with two substituents X and Y, the Hammett's constants could be expressed in the Hush equation as either K1 summation of sigma X plus sigma Y or K1 sigma X plus K2 sigma Y. Hush equations can be used to give information about the nature of the mechanism by which drugs act and as well as predict the activity of yet unsynthesized analogues. In the former case, the value of the constant for a parameter gives an indication of the importance of the influence of that parameter in the mechanism by which the drug acts. Consider, for example, a series of analogues whose activity is related to the parameters pi and sigma by the hypothetical Hush equation. Log 1 upon C is equal to 1.78 pi minus 0.12 sigma plus 1.674. In this equation, the small value of the coefficient for the sigma relative to that of pi shows that electronic factor is not an important feature of the action of these drugs. Furthermore, the value of the regression coefficient for the equation will indicate whether suitable parameters were used for its derivation. Values of R that are significantly lower than 0.9 indicate that either parameter used to derive the equation was unsuitable or that there is no relationship between the compounds used and their activity. This suggests that the mechanism by which these compounds act may be very different and therefore unrelated. Predictions of the activity of as yet unsynthesized analogues are useful in that they allow medicinal chemists to make a more informed choice as to which analogues to synthesize. However, these predictions should only be made within the limits used to establish that relationship. For example, if a range of partition coefficients with values from 3 to 8 were used to obtain an activity relationship partition coefficient equation, then this equation should be used to predict activities of those compounds only which have partition coefficient in this range. This means that this equation cannot be used to predict the activity of compounds with partition coefficient of less than 3 and greater than 8. Hush equation activity predictions that are widely different from the observed values suggest that the activity of the compound is affected by the factors that have not been included in the derivation of a Hush equation. The discovery of this type of anomaly may give some insight into the mechanism by which the compound acts. For example, a study by Hush on the activity of penicillins against a strain of Staphylococcus aureus in mice gave the in vivo relationship log 1/c is equivalent to minus 0.445 pi plus 5.673. This relationship predicts that a penicillin with a branched side chain should be less active than a penicillin with the similar sized unbranched side chain because the branched side chain has a higher pi value. However, Hush found that the penicillin with the branched side chain was more active. He suggested that this anomaly could be explained by the fact that branched side chains were more resistant to metabolism than straight side chains. Now coming to the accuracy and success of Hush model. The accuracy of Hush equations and hence the success of QSAR investigations depends on using enough analogues and the accuracy of the data and the choice of parameters. The greater the number of analogues used in a study, the greater the probability of deriving an accurate Hush equation. A rough rule of thumb is that minimum number of compounds used in the study should not be less than 5x, where x is the number of parameters used to obtain the relationship. Where the substituents are being varied, it is also necessary to use as wide a variety of substituents in as large a range of different positions as possible. The accuracy of the biological data used to establish a Hush equation will affect the accuracy of the derived relationship because its value depends to some extent on the subject used for the measurement. Consequently, it is necessary to take a statistically viable number of measurements of any biological data such as activity and use an average value in the derivation of Hush equation. Furthermore, extreme parameter values should not be used because they are likely to dominate the regression analysis and so give less accurate Hush equations. Their presence suggests that the parameter is either not suitable or there is no possible correlation. The choice of parameter is important because one parameter may give an equation with an acceptable regression constant, whereas another would give an equation with an unacceptable regression constant. For example, it may be possible to correlate successfully the dipole moments of a series of analogues with biological activity but fail to obtain a correlation for the same series of analogues using Hammett's constants as a parameter. Furthermore, some parameters like pi and sigma are interrelated and so their use in the same equation can lead to confusion in interpreting the equation. Lastly, coming to applications of Hush analysis. Quantitative relationships between the structure and the physico-chemical properties of the substances and their biological activity are being used as the foundation stone in the search of new medicines. The mathematical and statistical analysis like Hush analysis helps us to predict the drug activity. Hush analysis makes it easy to reach to the conclusion for any of the congener that still is not in process in a way that whether it will be optimal and profitable or not. To optimize the existing leads so as to improve their biological activities. To predict the biological activities of untested and sometimes yet unavailable compounds. So, to conclude with, we now know that to minimize the time required in drug formulation and synthesis, drug formulators predict the biological activity of the drugs prior to their formulation from their chemical structure analysis. Various mathematical models have been proposed to determine the biological activity of the drugs in vivo, but Hush's model is the most accepted one. It's based on the physico-chemical properties of the drug molecules like partition coefficient, electronic and steric properties. But its accuracy is very sensitive and depends upon the factors like the type of parameters, number of parameters used for its derivation. This is all. Hope you have understood well. Thank you and have a nice time.